This conference will now be recorded.
Hello.
Yes, sir. Share your screen.
It's I think some network issue is there. I cannot share my screen. Okay. Can you make me as a presenter?
This conference will now be recorded.
Okay. Am I audible?
Yes, sir. You are audible.
Okay. Fine. So today is good evening, everyone. So this is class 6, and today we are going to learn about your change the slide. Can you change the slide? Amit, change the slide. Yeah. So today we are going to learn about what is repetitive sequences, as we have already gone through C-value paradox and renaturation kinetics, and how this renaturation kinetics helps to find the different sequences which are present in the DNA. So we will then how this renaturation kinetics help us to find the repetitive sequences and non-repetitive sequences. So in this class we will go through about repetitive sequences, what is repetitive sequences, then different types of repetitive sequences, and then that there are clustered repetitive sequences, dispersed repetitive sequences, and um, clustered repetitive sequences has satellite DNA, mini satellite, microsatellite repetitive sequences, and dispersed repetitive sequences are moderately repetitive sequences, or you can say moderately renatured DNA sequences, which are usually a transposon molecule. So we will go through this dispersed repetitive sequences in eukaryotes, and what is gene. Then we will go through about what is gene, and then what is introns and exon in that, what is the function of gene, okay, and then what is the function of intron and what is the function of exon. Then what gene duplication, about gene duplication, and then what is the fate of gene duplication, how gene duplication uh, helps in evolution, or it helps to form gene families. We will learn in this class. Okay, next slide please. Okay, so we have already gone through in the previous class about this renaturation kinetics, or we can say as a Cot curve plot. We have learned this Cot curve and renaturation kinetics in the previous class, which tells that the different sequences which are present in the DNA has some sequences renatured very high rate, okay, and some sequences uh, renature at a moderate rate, and some sequences which are which take lot of time to renaturation. Okay, so this this type of things comes under renaturation kinetics. Okay, these techniques, what allows, it allows different classes of repeated DNA to be identified by the effect of multiple copies of sequence present on the DNA, and on the rate of renaturation of denatured genomic DNA fragment. Okay, by this method we have identified and classified, as I've already told you, we have classified as highly repetitive DNA, moderately repetitive DNA, and unique DNA sequences. Okay, so uh, this, in this way we have classified non-repetitive DNA and repetitive DNA, right? Can you change the slide? Please change the slide. Okay, so now repetitive sequences. So what are repetitive sequences and what is non-repetitive sequences? See, the fraction of DNA which reassociates very slowly, because it contains a unique sequences, usually having single copy in a haploid genome, right? So these sequences take lot of time to renature. If it if the DNA if these sequences get denatured and then we are renaturing it, so this unique sequences takes a lot of time to renature, because these are not repetitive sequences. It's a unique sequence and it's present in a single copy. Okay, and this single copy usually forms a protein part. Okay, this is the functional part. This single sequences, single copy gene sequences is the main, which produces a protein. Okay, this unique sequences is the base of our life. Okay, and this is known as non-repetitive sequences. Now what is repetitive sequences then? The fraction of DNA which reassociates or renatured faster as its renaturation rate is higher than the non-repetitive sequence. The non-repetitive sequence means unique sequences. Okay, and this sequences is called repetitive sequences because these sequences are repeated several times in the DNA, or you can say in the genome. Okay, so these sequences are repeated many times, so these sequences can renature very faster than the unique sequences. Okay, and as I told you, it comprises more than, obviously it is repeated many times in the sequence, so it will obviously consist of more than one copy of one copy, right? It will be present in more numbers in a haploid genome. Next slide. Please change the slide. Hello. Okay. Now this repetitive, I think, wait a second.
It is visible.
Yeah, slide is visible, but you people are change the slide which you are changing. It's very slow. Okay, no problem. I have opened in my laptop.
Okay, now the reason
Now,
is that Amit is facing a network issue. That's why the slide will be
yeah,
changed very slowly. Okay, just wait for
the same
1 second,
thing.
2 seconds, then the slide will be automatically. Yeah.
Okay, no problem. Fine. Okay, so now these repetitive sequences are differentiated in two forms, which is known as cluster repetitive sequences and dispersed repetitive sequence. So we will go through we will go through about clustered repetitive sequences. The name itself suggests that this clustered repetitive sequences, clustered means it will be present in an array form, or it will be present in one place or in one position of the uh, genome. Okay, and it will present in multiple times. Okay, it will be in a repeated form. So what is this cluster repetitive sequences? See, the human genome usually contains an extensive region, means it's contain a long region of highly repetitive DNA of very short sequences, which usually is about 2 base pair. It starts from 2 base pair and it goes to about 20 to 50 base pair in length, and that are arranged end on end as long tandem repeats, or you can say long tandem arrays in a form of arrays, okay, of many thousands of copies. In several copies it is present in the genome, and this type of sequences is highly repetitive DNA sequences, and it is called as satellite DNA. Okay, since they as why it is called as satellite DNA, I will show you one figure. In that you will find that, see, one you will get when you will run a density gradient centrifugation using a cesium chloride, then you will find that two bands will appear, in which one band having a higher larger peak and the another one is having a small peak. This small peak, the bigger peak is your main band DNA, okay, which is having all the sequence, and this the separate band which is having it tells about the your satellite DNA. Okay, I will show you in figure and then you will know. Okay, so this satellite DNA is identified by using a beyond your density gradient centrifugation using a cesium chloride when DNA fragments are being centrifuged. Okay, since this consists of repeats of such short sequences, so this renaturation usually depend upon the GC content. So this GC content will finally leads to the renaturation, and the renaturation rate also depend upon this GC content. Okay, and how long the sequences are being repeated. Okay, since this satellite DNA was the first to be identified in the human genome and contains repeats of up to 200 base pair, okay, and arranged in clusters between 100 to 5000 kilo base pair. It goes up to 5000 kilo base pair, means this repeat units which can be up to 22 to 200 base pair can be arranged in a cluster, which are present in a cluster in a genome. It can go through from 100 to 5000 kilo base pair. So it is present in a huge amount, right? So that's why it shows it's it shows a band when when we run a centrifugation, okay, beyond density gradient centrifugation. It gives a separate band, but when you will run centrifugation for mini satellite and micro satellite, you will not get, because their copies numbers are very less than the as compared to the satellite DNA. So that's the case. Now the example what we came to know about satellite DNA. Okay, so satellite DNA is having a repetitive sequences which are present in the genome, and it is consist of 200 base pair of repeats, and it is clustered between 100 to 5000 kilo base pair. Now what are the example? The examples are like alphoid DNA. Okay, these alphoid DNA are present near the centromere region, and if you find this alphoid DNA sequences, you'll find repeat sequences which are about 10 to 20 base pair long, even sometimes it consists of 200 base pair long, and it's repeated several times. Okay, now another example is rRNA encoding genes. Okay, these are RNA encoding like 45S, this ribosomal RNA. Okay, these are the 45S precursor. Usually 45S is synthesized in a precursor form. Okay, then it is been removed and then trimmed, and then it finally forms a 45S. It first synthesizes a 48S. Okay, this 45S is distributed in 18S, 5.8S, and 28S rRNAs, right? These sequences of these sequences which are present in the DNA for this rRNAs. Okay, these are present in a 10,000 copies in an repeated form. Okay, in a repeated form arranged in an array. So this the number of copies present in the present in the genome, it depend upon the species. Like in humans, it is present, the arrays are present in 5 separate chromosomes. Okay, five different chromosomes, these genes are present, and each containing 40 copies. Okay, and about how much long it will be, it will be along 100 or 200 base pair long repeats will be there, and number of copies in human is 40 copies. Okay, and it is present in 5 different separate chromosomes. Okay, so this is about, so now you can see in this figure, this is the experiment which is carried out to separate the this satellite DNA and the main band DNA. Okay, you can see the centric density gradients, and they are using a high concentration of salt, which is cesium chloride. Okay, and they have done density gradient centrifugation. So based on this density, the DNA is uh, can you change the slide? Yeah, so in I will explain again. So this is the experiment which is carried out to separate the main band DNA and the satellite DNA. So in the left side, you can see that the DNA is being denatured. Okay, and then they have done a beyond density gradient centrifugation, and then according to the density gradient, the DNA, so you can see the band has been appeared. Okay, now here you are getting one band. Usually you will get two bands which will show main band DNA and the satellite DNA if satellite DNA is present. So you can see in the right side, they have taken the mouse DNA, and they have done this cesium chloride density gradient centrifugation, and you can see according to the beyond density, it has been separated. The main band DNA is 1.7 gram per centimeter cube, and satellite DNA band, very small peak, you can see that is satellite DNA, which is coming in the 1.690 gram per centimeter cube. So in this way, you can distinguish the satellite DNA from the main band DNA. But if you, as I told you, if you want to find about whether now satellite DNA is further classified into two other type, that is mini satellite and microsatellite. So you can't distinguish this mini satellite and microsatellite on this way. It needs more advanced techniques, uh, uh, which you will learn in tools and techniques usually. So in this way they get separated, and finally you can see the two main band and the satellite band. Okay, change the slide please. So now based on this uh, density gradient centrifugation, there are two types of satellite DNA. One is mini satellite DNA and another is microsatellite DNA. Mini satellite DNA, which is also known as variable number tandem repeats, VNTR. These are usually used. This technique is usually used in fingerprinting. Okay, DNA fingerprinting. So, and microsatellite, these are simple tandem repeats which are usually present throughout the DNA. Now, what is mini satellite? So, mini satellite DNA occurs in small clusters up to 20 kilobase pair in length. Okay, satellite DNA, the satellite DNAs which were present in the DNA, it was about 100 to 500 kilobase per long. Okay, in present in cluster, but this mini satellite DNA is present in on up to 20, only up to 20 kilobase pair in length. So it is very smaller than the satellite DNA, and because of this reason, this mini satellite and microsatellite you can't see in that beyond density gradient centrifugation. Okay, so now the mini satellite DNA occurs in small clusters up to 20 kilobase per. Hello. Yep. So 20 kilobase per in length and has
Twinkle, it's a
repeat
humble
unit.
request to you. Please mute your mic. Twinkle, Twinkle, are you hearing me? It's a humble request to you. Please mute your mic. Okay, sir. Go ahead, sir.
Okay. Thank you, Arya. So this mini satellite DNA are present in clusters up to 20 kilobase pair in length, and it has repeat units of about 20 to 50 base pair each. Each containing 15 to 100 base
Sorry to interrupt you,
pairs.
sir.
So the
Once again,
repeat.
sir. Sorry to interrupt you, sir. Please, Twinkle, I request
Yeah.
you. Please mute your mic. It's a humble request to you, Twinkle. I think you are new in this platform. Please mute your mic for smooth conduction of session. Twinkle. Okay, sir. Now please continue. Sorry for inconvenience.
Okay.
Please go ahead, sir.
Okay. No problem. Yeah. So this mini satellite DNA is having a 20 kilobase pair in length. It has a repeat unit of about 20 to 50 base pair, and this each repeat unit consists of 15 to 100 base pair of sequences. Okay, the length of this particular simple sequence are present in array or is in cluster form, and it varies individual to individual. Because of this this mini satellites or VNTRs are being used to distinguish one person to another. Because of this variation in individuals, even in species to species also, even in interspecies also. Okay, so now, and this differences occurs because of the unequal crossing over during meiosis. Okay, these differences in length of mini satellite DNA forms the basis of DNA fingerprinting. Okay, now another example of the example of mini satellite DNA is the telomeric DNA. You will find that at the end of this the chromosome, the telomeric DNA sequence is being repeated several times. Okay, and it is repeated several times. It is present in 100 copies, and the sequences is what? TTA GG triple G. Okay, this sequences is present in 100 times, are repeated 100 times at the end of the chromosome, and this is telomeric DNA sequences. Okay, so this is the example of mini satellite, and usually it is present near the end of the this mini satellite DNA sequences are usually present in the near end of the chromosome. Okay, now microsatellite DNA. Microsatellite DNA occurs it is smaller than the mini satellite satellite DNA, very small than the mini satellite DNA, and usually present in about only 150 base pair, and only 150 base pair. It's present in 150 base pair cluster form, and has a repeat unit of 4 base pair, 2 base pair, or 1 base pair. Okay, and maximum it is where the repeat unit will be 4 base pair. Okay, and the repeated this repeat units will be repeated 10 to 20 times in the genome. So microsatellite DNA is very common because it is present throughout the chromosome. It is evenly distributed throughout the chromosome, and for example, one type of your repeat, if you'll see in the human genome, you will find that CA is the dinucleotide which is repeated most probably in the entire genome, and it is present in about 0.5%, okay, of the whole sequence. Likewise, one mononucleotide sequences are also present, is present in about 0.3% of the whole genome sequence. Now, as I already told, that mini satellites are more frequently present near or end of the chromosome, while microsatellites are more evenly distributed along the chromosome, and throughout the chromosome it is been distributed. Okay, but it is present in the clustered form. Okay, this dispersive repetitive sequences are also present in the uh, throughout the chromosome, but they are not clustered. They are randomly, they are not present in cluster form. They are present, their position will be different, and their chromosome position will be different, chromosome number will be different. Okay, please change the slide. Okay, so this is the table. You can see, like satellite DNA usually it is present in the cluster form of about 100 kb to several MB. Okay, like 5000 kb maximum. Okay, and then this repeat unit size is 5 to 171 base pair, mainly located at centromere position. Now this satellite DNA is further being distinguished as alpha satellite and beta satellite, and this alpha satellite means alphoid DNA. One of the alphoid DNA, it is alpha satellite and beta satellite. These are present in centromeric heterochromatin region. Okay, and this size of the repeat unit is 771, and for alphoid satellite DNA and beta satellite DNA has a repeat unit of about 68. Okay, now this mini satellite DNA size, the cluster size is up to 20 kb, and the size unit is about 9 to 64 repeat unit. And as I told you, it is present near or end of the chromosome, like telomere DNA sequencer. Okay, now microsatellite, it is less than the 100 base pair or 150 base pair, you can say, and the repeat unit is a maximum of about 4 base pair, 12 base pair. Okay, may not more than 13 base pair, but it will be less than 13 base pair, the repeat unit. Okay, and this repeat unit will consist of about two base pair or 3 base pair or 4 base pair. Okay, not more than 4 base pair. Yeah. Okay, now this microsatellite is usually dispersed throughout the chromosome. Next slide. Okay, now dispersed repetitive sequences. This dispersed repetitive sequences are usually a moderately repetitive sequences. These moderately repetitive sequences are short sequences that are repeated typically 10 to 1000 times in the genome. Okay, these sequences are dispersed throughout the genome, as the name suggests that it has been distributed throughout, but it is not present in the cluster form. Okay, now this repetitive sequences is known as moderately repetitive sequences because it is the renaturation rate is um, slower than the highly repetitive sequences but higher than the unique sequences. So that's why its name is moderately repetitive DNA sequences. These moderately repetitive sequences usually consist of mobile DNA elements, or you can say as a transposon or jumping DNA. Okay, or selfish DNA also, you can say in that case. Okay, this DNAs has the ability to move from one location to the another location. Okay, so now in eukaryotes, these are usually present in the present as signs and lines. Okay, short interspace sequences and long interspace nucleotide sequences. Okay, now the best example of this sign is ALU element. Okay, this sequence are not identical, but they are similar enough to be classed as a family. Okay, this can be placed as ALU elements mean they are like, because this sign sequences has an this ALU is a restriction in them. So this sign sequences, this sign repeat sequences has a ALU recognition site. Okay, means restriction site. So it comes under the family of ALU family. That's why it told us that these sequences are not identical, but they are similar because of the presence of ALU elements. Okay, ALU sequences. so that they comes under the ALU family. Okay, so now they have an average length of 250 base pair, and it's present almost around 1 million copies. So it is so huge that it is present throughout the chromosome, and it is the copy number is 1 million copies. So it's very large and widely distributed throughout the genome, and it usually present in the form of introns in some genes. Okay, now the origin of this ALU element is uncertain, where from this element where it comes. Usually it believed that it has been pseudogenes because it has no function. Only it usually it can't jump, it does not able to jump itself. It has to depend upon the line to get this transposase enzyme. It does not synthesize itself a transposase enzyme, so it does not possess any activity, therefore it thought about that it's a pseudogene. Okay, it behaves like a gene, but it does not have a proper function, or it does not itself act on it. It depend upon the other. That's why the name given as a pseudogene. Okay, now this suggested that at this stage these sequences acquired transposable activity. As I told, it depend upon the transpose enzyme uh, which is synthesized by lines. Okay, and then it can move from one site to another site in the genome. Okay, next slide. So now this is another your dispersive or transposable element which is present in the eukaryotic genome. It is similar to SINE, but it has longer sequences and it has its own transposase enzyme. So it has its own activity of getting replicated and inserted into a genome from one side to another. So this is the and the second difference is that it is longer. Okay, like the example of line is L1 line element. Okay, like in SINE it was the ALU element. So in line it is L1 line element, which is 6500 base pair long, and it is present as 60,000 copies. Okay, 60,000 copies less than the your SINE sequences. Science were present in 1 million copies, so it is lesser than that. Okay, lines are type of retro element, as it can it can synthesize two enzymes. One is reverse transcripted. Okay, and then after reverse transcription, it synthesizes transposase enzyme, which helps to incorporate the sequences into the another site of the genome, or to another location of the genome. Okay, so lines are type of retro element. These are sequences that are capable of moving through the genome by a process, and this by a process called transposition. This movement from one place to another is called the process is called as transposition. Okay, which allows them to copy themselves by reverse transcription and to insert the copy into the genome at the distant site. So now most L1 elements are usually truncated. Truncated means it is been break down. Okay, but the full length version has all the component needed for transposition, including a gene that could code for reverse transcriptase. Okay, next slide. So now what is gene? So we know that gene resides in the DNA, right? So what do you understand by gene? A gene is what? A gene is the basic physical and functional unit of heredity. This is the definition which usually we are learning from class 10, 12, BSc, MSc. So what is gene? A gene is the functional unit of life, or is the physical and functional unit of heredity, right? Now the primary function of the genome is to specify RNA, right? So each region of the DNA helix has to produce a functional RNA, and this functional RNA is synthesized from where? Is synthesized from gene, and gene is present in where? Gene is present in the DNA. So these genes act as an instruction to make, and then finally these RNA sequences or gene sequences are finally gives instruction to make what? It gives make function to make a protein, and these proteins helps to regulate our body's metabolic process and one of the functional unit of life. However, many genes usually have been found that do not code for proteins. Okay, so excuse me. In human genes vary from size to size, from a few hundred DNA bases to more than 2 billion bases. But this size does not depend upon the sequences which synthesizes protein. It depends upon the sequences which does not synthesize a protein. Okay, and this make the size of the sequences which are which is called as introns. Okay, that makes the variation in the gene size. Okay, even in species to species, even in interspecies. Now the Human Genome Project has estimated that humans have 20,000 to 25,000 genes. Excuse me, wait a second. Okay, so as we have came across in the previous class that the Human Genome Project have given some silent features of human genome. Okay, so the human genome consists of 20,000 to 25,000 genes. The smallest now, as and the gene size vary from species to species also. So the smallest known genome is of the which has less number of genes is a Mycobacterium genitalium, has only about 470 genes, while humans having 20,000 to 25,000 genes. Now every person has 2 copies of each gene, one inherited from each parent. Okay, most genes are same in all people, but a small number of genes, less than 1% of the total, are slightly different between people's. Alleles are form of the same gene. Okay, so the alleles are present on the form as present on the gene, and it produces a small differences in their sequence of DNA bases. Okay, these small differences contribute to each person as a unique physical features. Like some people will have a red eye and some people will have a slight brown eye. Excuse me. So these features come because of this 1% differences in the sequences of the gene. Okay, next slide. Now the size, as we came across, the size of the genes in higher eukaryotes vary greatly. Okay, some genes contain more than 1 million nucleotide pairs. However, genes that are more than 100,000 nucleotide pairs in length are mostly present in commonly present in the genome, and this most of the portion of the gene in higher eukaryotes consist of non-coding DNA, like that interrupt the relative short segments of coding sequences. This coding information of a gene called as an exon, and this intervening sequences, or you can say like interruptive sequences, which does not code for protein, is called as a introns. Okay, the coding. So now the RNA molecule which are usually synthesized, which is called as primary RNA transcript, is synthesized is synthesized as a precursor RNA molecule has introns and exons. So we know that introns does not code for protein, only exons will code for protein. So this intron has to be removed, and the process by which these introns are being removed is known as RNA splicing. Now So now what exons we will come to know about the exons more deeper when we'll read about transcription. Okay. But introns we have to. Now I will give the details about intron. Okay, introns were first discovered in 1977 by Philip and Richard. Okay, these two scientists had discovered the introns present in eukaryotes genome in a complex manner. Although they are not universal, like many genes in the human, like interferons, histones, and heat shock protein, G protein, uh, ribonuclease, all these proteins lack intron. So from this you can tell that introns have no functional role, and or it does not require the function of presence of intron. uh, Even the introns are not present, then also gene will function in eukaryotic cells. So in addition, genes are not introns are not bound in most of the genes of simple eukaryotes like yeast. In yeast you will find that there are very less number of introns, or rare introns are present. And likewise in prokaryotes, introns are rarely present in the gene. Okay. Now but the large gene consists of a long string of but in eukaryotes the larger gene will consist of a introns part, and it will be present in an alternating form: intron, then exon, then intron, then exon, likewise. Okay. So with the most of the gene consisting of introns, the over length of the gene, as I have told you, the length of the gene will not is not determined by the exon part, but it is determined by the intron part. Okay, introns range in size from about 50, for example you can say, like the intros if has 50 nucleotide to 80,000 800,000 nucleotide, then usually exons in this case it will be very less. It will be likewise it will be in 150 nucleotide only. So introns is in huge number, but exons are in less number. So this intron has to be removed. It does not have any uh, important function which will help to synthesize protein. But in some cases it has been found that it plays role. It plays role to attach the kinetochore in the centromeric region. Such function has been found, but it does not produces any protein, or it does not completely functional as an exon part. Okay. Next slide please. So this is the experiment which is called as Rloop technique. This Rloop technique is used to find the introns. Okay, the scientists have using this technique they have identified that okay, the gene is also having a intron part. It is not only that the gene is not only the functional or exon, only the exon sequences are there. It has some non exon sequences which does not produces any protein. So in this technique usually what they did, they they have taken they have taken one mRNA molecule. Okay. And then the DNA molecule, and they have annealed this single stranded mRNA and double stranded DNA molecule, and then they they visualize in the electron microscope. Once the RNA DNA base pairing happens and it forms RNA DNA helix, then they found that the exon parts are being attached with the DNA sequences. Okay, the mRNA is what the mRNA, the synthesized mRNA. This is not the precursor mRNA. This is the functional mRNA, which is only having the exon part. It does not have any intron part. So this exon, this the sequence is only having an exon sequences, right? So these sequences, when it's matching with the DNA sequences, it is getting base pair, and the introns part are not getting base pair. It forms a loop structure, large loop structure. This large loop structure is only the intron. So this experiment helps to find out that no, gene is not only having an exon, it is also having an intron. Okay, next slide. Now we will go through about gene duplication. So what is gene duplication? So gene duplication is the process by which a region of DNA coding for a gene is copied, means it has been duplicated, it has been multiplied. Okay, and this gene duplication usually occurs in occur as a result of error in recombination or through retrotransposon or unequal, you can say, recombination on the basis of unequal crossing over, or in the case of retrotransposition or by the replication seepage, means error in the replication process. Okay, now that these duplicate genes uh, are often selected by a selective pressure under which genes normally exist, means one gene has been lost its function but it has been copied to another position of the DNA, but this gene now the original gene was mutated but the copied gene is still existing. So the mutated function will not, the lacking of that gene will not happen. The functional protein will still synthesize even if the original gene sequences has been mutated, because the copied gene is already present. Okay, so this is called a selective pressure. Okay, still the uh, mutated gene is present, but because of the copied of that sequences present in another location, it still synthesizes a functional protein. Now this can result in a large number of mutation, but if it keeps on going like that, then it will obviously produce a large number of mutation, and finally it leads to cause a disease, or maybe it will cause a non-functional to that gene, but it in some cases it gives benefit to the organism. Like you can say you can say it helps in it helps in synthesizing families of protein, like globin family protein, beta globin, alpha globin. So these are synthesized, only few sequences has been changed. um, the sequence is completely similar, only few regions of the sequences is being changed, but the function but the function of that protein is distinct, is different from the alpha globin, and alpha globin will function in different way and beta globin will function in different way, but the sequence is almost similar, but the position of that sequences is different. Okay, this is the result of gene duplication. Okay, next slide. Excuse me. Now, the mechanism by which this gene duplication occur is ectopic recombination. Ectopic recombination means error or error recombination. It's not a classical recombination. Recombination process usually happens in uh, like crossing over event. Okay, it is also taken care of by the cell by using several enzymes. If it requires a repair, it undergoes repair, but sometimes this ectopic means unequal crossing over result in gene duplication. Now replication slippage means the error in replication process can also result. Non-retrotransposition, as we have learned about line and sign, this also result in gene duplication. And then aneuploidy, which chromosome number increase, that is also a type of a gene duplication. So these are the process through which gene duplication happens. Okay, now the fate of duplication. What is the fate of duplication? like I have already told you that it helps in evolution. It it has it is used to find the evolutionary relationship of the organism. Because if the organism is coming from one species, suppose it's 100 populations of organism, 100 organisms are there, and four of same species, and 10 of 10 are evolving because of this duplication. So this duplication, but still they are surviving, uh, still they are surviving. So still has its own this because of this duplication, but it shows a different trait. Okay, this this is called a selective pressure. Okay, and this pressure gives a different organism or different trait, but it has come from the same ancestor or same species. Okay, so the first fate of duplication is that in which the most the in most of the cases it has been found that there is a loss of function of mutation which functionally restore the one gene state that preceded the duplication. And as I told you that the original gene has been mutated, but because of duplication, that particular gene is present in the another location of the DNA. So the gene comprise the gene still functions. Okay, it gives the functional protein. So that is the first case. Okay, even if it is getting mutated, no problem. I have one more copy of that. So that helps, it gives a beneficial to the organism sometimes. But if the loss is, if the duplication occurs completely in the exon region and the incorporation of that sequences is in intron region, then the mutation is harmful for the organism. Now the second fate of duplication is what? In this the duplicated gene remains functional. Okay, the gene is functional, but the sequences are having a divergence from the previous from the parental sequences, and this led to a different physical properties. And this is and finally it gives a different family of genes, as I have given the example like myoglobin, hemoglobin, alpha and beta globin protein is a globin gene family protein, and this is because of gene duplication, because gene duplication and divergence. Okay, uh, next slide. So you can see here, so this is gene A having a sequence of A, C, T, C and T, G, A, G. Okay, now gene duplication process happened. Now you can see the selective pressure happened. Okay, in this selective pressure, the still uh, even this gene A, the original gene sequence is mutated, but it having a again the sequence is having a functional properties and it perform a original function. So there is no loss of mutation, no loss of mutation. Okay, even if the original sequence is not working, copied one is also present, so it gives a functional protein. Now in the second case, you can say in the divergence case, okay, ACTC it's been changed to GATG and CTAC. Now this gene B is again synthesizing as GATG and CTAC, but in the next sequences it is synthesizing as a GATTG and CAAC. Okay, so the first one is producing a related function, but the other one is not producing a related functional protein. It's a pseudogene, it's silent. Okay, so likewise if you'll see the protein sequences of alpha globin and beta globin, you will find in similar manner there is some slight sequences which makes this protein distinct, but they are the but almost 99.9% sequences is similar. Okay, and this is because of the gene duplication and divergent process. Okay, so the next class we will go through about homologous gene, what is homologous gene, and then pseudo gene, and then finally then we will learn about transposition process and all transposable element what in bacteria and eukaryote. So I end up here. So thank you to all for listening.